Before a cell goes to make another cell, it needs to make more DNA because each cell needs a full set of instructions. And that process is DNA replication. You know, back in 1953 when Watson and Crick described what DNA looked like, came up with a model, something like that, they also described how they thought DNA copied itself. And they're pretty dang close. Since then, we've learned a huge amount of information. You could spend an entire class just studying this process. In this video, I just want to touch on the basics, the idea of DNA structure, complementary base pairing, and the enzymes used, at least the major ones. So with that, let's get into it. So here's the chemical structure of a nucleotide. Pretty complicated, huh? Let's break it down because it's not as bad as you might think. So every single one has a phosphate group. And phosphates are things we're going to see again and again. A lot of times, instead of writing all that, we just write a P with a circle around it. Sometimes you'll even see a P with a little I by it, meaning inorganic phosphate. Next, we have this sugar. This one is a deoxyribose sugar. And then finally, the most important part is this nitrogen base. And there are 4 different nitrogen bases. Let's take a look at those. So here they are. You'll notice that we, in each of these, again, have the phosphate and the sugar. But in this case, we have this one for adenine. And I usually just refer to them by their letters. So just A. This one is T. This one is C. And this one is G. That's it. A, T, G, and C. All right, so those are the 4 monomers that get put together. It'd be like these different colored cups. So this might be the A, and then T could go with that, and the G, and the C. And they only go together kind of in one direction, but they're all somewhat interchangeable. They just go together in a line. We also can symbolize it when we write it just with the letters. Those sugars and phosphates don't matter a whole lot, so a lot of times we just draw it like this, just those nitrogen bases. Sometimes you'll see it something like this, indicating there's that sugar phosphate backbone. And these are the nitrogen bases in the middle. So there would be one of our strands. Let's see what that would look like in a little more realistic chemical perspective. Here are a couple of different representations of the DNA polymer. Let's look at this one on the left. It's a little more simplified view. We have this blue ribbon here being the sugar phosphate backbone. And notice that there are 2 of them. So there are 2 strands of DNA ranged in this spiral pattern. And this is often referred to as a double helix. So this is a big, big molecule. And on the inside of those bases, those nitrogen bases, the A, T, G, and C, just symbolized by colored rods in there. So on the right side, we have a more complicated view. We have those same sugar phosphate backbones that we're going to kind of ignore for now. And we also then have the bases, thymine, adenine, cytosine, and guanine. So there they all are in all their chemical glory. But this is just so much to take in. This one on the left is quite a bit simpler, but it doesn't give us quite enough detail to understand it fully. So a lot of times we kind of strike a balance between too much detail and too little. So there's the DNA double helix. Let's zoom in on the details of how these 2 strands go together, focusing on complementary bases or complementary base pairing. So DNA is double-stranded. Here I have an orange and a white strand together. So here's the detail of the orange strand. The white strand is actually really predictable because, as you noticed on those other diagrams, there's always one nitrogen base opposite the other one. And so the one opposite here is its complementary base. And the rules for that are simple. A and T always go together, and G and C always go together. I like to think of it as straight line letters go together and curvy line letters go together. So these aren't matching, they're complementary. Kind of like certain shirts go with pants. This might be one outfit and this is a different outfit, but you can't really mix and match them. So let's apply it to this. Complementary to A is T, and complementary to T is A. Now that you got that, you can probably do the rest yourself. This is pretty simple. There you go. That's it. We already know what the other strand is going to be. And this actually suggests how DNA copies itself. Well, so far we know what. It involves nucleotides. We know when, right before cell division. We know where, wherever the DNA is. For us, that's in our nucleus. And now on to how. How does this happen? To give you a bit of a preview and an analogy for this, DNA replication, that copying process is different than ours. So if we had a fantastic work of art like this that we wanted to make a copy of, then we would either draw a new copy or we take this, put it in a copy machine, and pull out a new copy. But that's not how DNA replication works. So DNA is double-stranded, not like this. So now we have a document more like DNA. And if we wanted to copy it, first we'd have to pull it apart and just work on one strand at a time. This I would put into the DNA copy machine, and what I'd get out was the other half. It would complete it for me. And I could do the same thing with the other half, put it in the DNA copy machine, and it would give me the other half. Pretty strange. So don't think of it as copying like you would see something and you would copy it down. It's a very different process. Let's take a look. Now, one last piece. First, we need to unwind and separate or unzip this double helix. This is a pretty twisted up molecule. That job is done by an enzyme known as helicase. It deals with the helix of DNA. And then we need to add these new strands, these yellow ones down here. And so that's the job of DNA polymerase. It makes DNA polymers. Simple enough. So those are the two molecular tools we'll deal with in this one. I also want to note there that we had two strands here in blue, and down below we have one of those left and one left over here. So each of the new double-stranded DNA complexes has one of the old strands. And that's why it's called semi-conservative. Semi because only half is conserved, and conservative because we kept or conserved one of those strands. So it's not like we just copied it and then destroyed this old strand. Let's look at just a little more detail about how that strand is made. We've already revealed most of these secrets, but DNA polymerase, its job is to come in here and guide these nucleotides toward their complementary bases and then bind them together. So you notice how all these have that sugar phosphate backbone. And so now that G right here is going to be added along with the next G, and we'll form a covalent bond with the previous one. Then we'll add another G and a T and a C. You get the idea. That complementary base pairing is pretty simple, and DNA polymerase has a really important job of making sure exactly the right nucleotides get added that are complementary to the old strand. So there's the basics of DNA replication. Not too hard. DNA copies itself using complementary bases. That's a good foundation for you to start from. Make sure you understand this before you move on. I've not tried to animate any of this. There's so many great animations out there for you on the web. I've given you some links to them. Take a look at those. It does make it come to life a little bit more, understanding how this works. It's an amazing process, and it happens every time before your body makes a new cell. Now we could fill up books and books of information on the rest of the details. In the next video, I'll add a few more enzymes and a little more detail onto the rest of the process, because it is a little more complicated than just what I presented here. But this is a solid foundation. My suggestion is, before you move on to that video, that you try explaining it for yourself. Write out a short paragraph describing this. Explain it to a friend. Make sure that you have the foundations down before you move on. Until next time, keep being amazed by the intricacies of life.